Avery, MacLeod, and McCarty used principles from Griffith's experiment to figure out what was a transforming factor in cells, whether it be RNA, proteins, or DNA. They achieved this by heat killing virulent bacteria and then adding it to test tubes containing RNAs, protease, or DNAs. These test tubes were then added to flasks containing non-virulent bacteria. These non-virulent bacteria were transformed into virulent bacteria in the RNase and the protease flasks, but the DNase flask remained non-virulent, therefore proving that DNA was a transforming factor. Hershey and Chase's bacteriophage experiment sought to discover what part of the phage was the genetic material that was transmitted into the host. Was it the protein or was it the DNA? They grew bacteriophages in radioactive sulfur for the protein and radioactive phosphorus for the DNA. They then allowed the bacteriophages to infect E. coli in a separate flask, using them as hosts. After some centrifugation and blending, it was discovered that only in the phosphorus sample was radioactive cells found. In the sulfur sample, the radioactive material was found in the liquid. This proves that DNA, not protein, was the genetic material for bacteriophages. Messelson and Stahl wanted to know how DNA was replicated, so they grew E. coli in a heavy nitrogen media so the DNAs would be filled with heavy nitrogen, and then they allowed it to replicate 3 times in a normal nitrogen media. The DNA was extracted from these 4 samples and then centrifuged in a tube. The heavy nitrogen sample produced a band close to the bottom of the tube. The first round of replication produced an intermediate band. The second round produced a light and intermediate band. The third produced a thicker light band and an intermediate band, thus giving evidence that it was semi-conservative replication. Taylor, Woods, and Hughes also sought to figure out how DNA replication occurred. They used autoradiography. They allowed one cellular division in the presence of radioactive thymine so that both sister chromatomes would be radioactively labeled, and then they allowed another cellular division in the presence of normal thymine. What they found was that one sister chromatid was radioactively labeled while the other was not, proving that replication was a semi-conservative process.